We continue with the search for genetic material. This is video, that is part 3 for the topic. Part 1 we discussed Griffith's experiment, coming to the transforming principle. Part 2 was a video by experiments done, covering the experiments done by Avedi, McCarty and McLeod, who quite genuinely proved that DNA is the genetic material, but since they could not provide any convincing evidences, many scientists were not accepting this fact. So the debate went on. Actually, the period between 1940 to 50 was continuing with a serious debate on what exactly is the genetic material, whether it is protein or DNA. Of course, finally it was between protein and DNA, most of them believing that protein is the genetic material. As I said in the previous video, chromosomes were not given so much of importance. So even their experiments did not convince many people. It was in the year 1952 when Alfred, Hershey and Martha Chase further carried out experiments to sort out this puzzle, whether protein or DNA was the genetic material. And so they selected a simple model organism, that is a virus particle, that is Bacteriophage T2. Bacteriophage, the name is bacteria eater, phage meaning eater. That means this virus particle infects bacterial cells. They grow inside the bacterial cells. They propagate inside the bacterial cells, destroying the bacterial cell. So it is said they are eaters, bacterial eaters, bacteriophage. T2 is the type 2. There are T2, T3, T4, T5, etc. like that. So this is T2 phage particle, type 2. So why did they select this model organism, bacteriophage? And of course, bacterial cell, E.coli was also selected. This virus infects bacterial cells, that is why. And most importantly, this virus has only two molecules, DNA and protein. That is the main reason why they selected it as their model organism. Another important question, what was the basis of Hershey and Chase experiment? Since this has only DNA as a core molecule surrounded by protective protein coat, it was very easy for them to label DNA and protein in this organism, in this virus. So DNA contains phosphorus, we know, on its phosphate backbone. It does not have sulfur. DNA does not have sulfur. On the other hand, proteins have sulfur in some of their amino acids like cysteine and methionine. But proteins do not have phosphorus. So that was very clear. DNA contains phosphorus, which is absent in protein. Protein contains sulfur, which is absent in DNA. So labeling phosphorus in DNA and labeling sulfur in protein, that is tagging for radioactivity, was very easy. So they selected this organism, and that was the basis, that labeling being possible due to this components missing, one component missing in each, and only one component present in each, phosphorus in DNA and sulfur in protein, was the basis of this experiment, why they selected this. Okay, so before we start with the experiment, let us see how the bacteriophage attacks a bacteria and destroys it. A phage particle, when added to bacterial culture, they adsorb to the bacterial surface. They will adhere there. And they will inject some material in. Here see, we know that it is DNA being injected, but at that time it was not known what exactly goes inside. Bacteriophage attacks bacteria, grows inside bacteria is fine, but what was it? Was it protein or was it DNA that was injected in, was not known. So basically we'll understand the cycle first. So it injects some material inside, and that material is treated, that genetic material gets incorporated into the bacterial genome, and the bacterial cell treats that viral genetic material as its own and starts manufacturing more and more virus particles. That means the genetic material which enters from the virus into the bacteria makes the bacteria obey it and makes components for itself, not allowing bacterial components to be formed. So gradually the bacteria becomes weak and lysis occurs. This is the lytic cycle of bacteriophage, and many progeny virus particles, newly formed virus particles are released, which will again infect new bacterial cells. So this is the life cycle basically of a bacteriophage. In 20 minutes of infection and all this assembly and all, they will burst. Bacterial cell will burst, releasing all the progeny. Here it is a view of how the phage particle adheres to the bacterial cell. So they started their experiment with the preparation of two different types of radioactively labeled phage particles. They wanted two populations of bacteriophages, one with radioactive DNA and the other with radioactive protein. So what did they do? They cultured bacterial cells in phosphorus 32, P32 isotope. Normal isotope of phosphorus is 31. They cultured bacterial cells in such culture plate with nutrient medium containing P32. They allowed, I have just highlighted one bacterial cell here, they allowed bacteriophage virus to infect the bacterial cell, so that, as we discuss now how the cycle goes inside, and after bursting of the cell, after lysis of the bacterial cell, they could get progeny viruses which were all having DNA radiolabeled. So, before we understand, let us see what is radiolabeling. What is isotope? You know, isotopes are two or more forms of the same element. So here we have the normal P31, we are taking P32 which is radioactive. P31 is stable, P32 is unstable, and so it is radioactive. Normally, routinely, P32 is used in laboratories to produce radio-labeled DNA because it is very readily ingested and incorporated into the nucleic acid. So definitely the bacterial cells which were consuming the nutrient medium containing P32, they got P32 in their phosphate backbone. And when the bacteriophages were growing inside those bacterial cells, they were incorporating it from the bacterial cell. They were getting it from the bacterial cell. So their DNA also was labeled because of that P32 present in the nutrient medium consumed by bacteria and finally incorporated into the viral progeny. So they got a population, the scientists got a population of labeled DNA phages. Similarly, in the culture plate containing S35, this was meant to label protein because sulfur is a component of protein. So they got bacterial cultures in it. Bacteria incorporated this S35 in their protein. So when phage particles were allowed to infect, they also got their protein code having S35, which would be radio-labeled when radioactivity would be studied. And this red and green color has nothing to do with it. This is just for understanding. What is radio-labeling? Radioactive components, they emit radiation in the form of particles. Substances that contain unstable atomic nuclei is radioactive. Like I said, P31 is stable, P32 is unstable. Likewise, here sulfur 35 is unstable. So they will emit radioactivity. They will emit radiation. So there is a way of measuring the radioactivity at the end of the experiment. So that is one reason why they selected these two types of culture is to label each component so that each of these components in DNA and protein would be identifiable when recovered at the end of the experiment. So they started with these two populations of the bacteriophages. One population with DNA labeled and the other population with protein labeled due to S35 in it. So what was the next? Their experiment began with the first step called infection. Each of the two populations of radio-labeled phages were allowed to infect fresh E.coli cultures. Now the culture plate was not given radioactive elements. Normal phosphorus and sulfur was incorporated with the other nutrients in the media. So when these freshly cultured bacterial cells were infected with our radio-labeled phage particles, what happened? That was the question. So allowing, this is the E. coli normal view that we see. This is just a picturization of the diagrammatic representation of what we are studying. This is how the coli looks, colony of the E. coli looks. So when the bacteriophages were allowed to infect fresh bacteria in the fresh culture medium, definitely they injected something. What did they inject? That was the question. Was it the P32 containing DNA that went in, or did that P32 remain outside? This we had to check. This would be highlighted with radioactivity when checked. If it is outside, it will show radioactivity outside the bacterial cell. If it is entered into the bacterial cells will show radioactivity. Similarly, this case, whether it was the protein code that entered or whether it was protein code remaining outside the cells. So if it was remaining outside, the solution outside the bacterial cells would show radioactivity, and if it is inside, the bacterial cells would show radioactivity. This is the puzzle to be solved. So second step was agitation in a blender. So in separate blenders, both of those populations solutions were blended, agitated. That means they were a little bit shaken, so that whatever adhered to the bacterial cell would move away from the cell. It will detach from the cell. So sheared off PHAGE coats we mentioned here, because today we know it is a coat that was removed off. So whatever was adhered, what went in would remain in the bacterial cell. What was adhered on the surface would be disconnected, would be detached. For that, blending is done. So what is the function of blending? To remove off the empty PHAGE coats, not to separate, to remove off from the bacterial cell surface. Then the next step is done. So this is called shearing off of the PHAGE coat. Next step is done called centrifugation, where the two molecules DNA and protein would be separated off completely. So that is a low speed centrifugation done called spinning. When it is done in a low RPM, rotations per minute, what happens is the heavy particles, the bacteria being bigger, will centrifuge to the bottom and collect as pellets, whereas the smaller particles, virus being smaller, will remain in the supernatant liquid, floating in the supernatant liquid. This is what happens on centrifugation. So the top part is called supernatant and the settled part is called pellets. So bacterial cells would be in the pellets. So when radioactivity is measured, now this is the point where you measure radioactivity. You will see in the first sample population of the PHAGE, where we had labeled the phosphate P32, okay, this 32 should be here, okay. That particles will not show, that means sulfur was not radiolabeled. So this supernatant will not show radioactivity because we had not labeled sulfur, we had labeled phosphates. Phosphorus was isotope radioactive. So this will show radioactivity. Whereas in the second population, radioactivity was seen in the supernatant because sulfur is a component of protein. So it is very clear that supernatant contains the protein. Whatever was inside the bacterial cells, that is showing radio labeling only here. Radioactivity is all shown in the bacterial cells, in the pellets. That means DNA has entered the bacterial cells. Whereas here, no radioactivity inside the bacterial cells. We had not labeled it with radiolabeled P. So definitely that has entered inside the bacterial cells. So the sheared off coats are separated from the infected bacterial cells due to centrifugation, and radioactivity is measured, indicating that DNA of the PHAGE entered the host cell, whereas protein coat remained outside the cell. And what is the conclusion? DNA is the genetic material and not protein. The DNA that goes inside confers its ability to produce progeny viruses inside the bacteria. Thus, protein has no function in its growth. Protein's function is to protect the DNA when the particles remain isolated. So, that is how it was concluded finally, that DNA is the genetic material. Moreover, on lysis of those bacterial cells, it was found that these bacterial cells also contained progeny, most of them having radioactivity shown. The progeny phages also were labeled in this case, whereas the progeny phages here were with having radioactive sulfur in them. In this bacterial cell, when brought out, when the progeny cells were checked, they were not having radiolabeling. They did not show any radiolabeled substance. That means that protein was not the genetic material. Instructions were not given by protein. If instructions were given by protein, then sulfur also would be incorporated in this protein coats of the progeny. So instruction is given by DNA. Also, the viral progeny is formed inside the bacterial cell. That also is a proof that genetic material was inside the bacterial cell. That's all. Thank you. With that, it is confirmed that DNA is the genetic material. Keep watching and keep reading. Thank you.